Good afternoon, good morning for some of you, maybe good evening for others. My name is Alex Raymond. I'm a professor at the University of Lausanne in Switzerland, and I'm also co-chair of the Scientific Program Committee of the European Society of Human Genetics. And because of this, I will use a few minutes of your time to advertise our upcoming conference, ESHG conference in Milan, from Saturday, May 24 to Tuesday, May 27. So 4 days of human genetics, of great science. We have 110 invited speakers, and we just a week ago selected 204 abstracts for platform presentation. And for the people who cannot join us in Milan in person, for the 4th year in a row, we will be fully hybrid. So you can join as you are joining here online and participate and follow the different scientific presentation in Milan. And this, we put a really low flat fee to have as many people having the possibility to participate. And the flat fee will be €100 in Europe, €50 for students, European students. And this flat fee will be even lower for people outside of Europe. And so please, catch this opportunity to hear about great science. Something I should say on a logistical point of view, you see that there is a chat, but there is also a Q&A function. So please use the Q&A function to ask questions you want us to relate and to transmit to Nicky. So while we have our annual conference, Bill Newman, our president, decided that we should use also the possibilities, technological possibilities today to give you and speak about and present great science throughout the year. And this is why we have a second episode of this ESHG webinar. And we have the great chance to have Nicky Whiffin with us. Let me tell you a few words about Nicky. She's associate professor and group leader at the Big Data Institute and the Wellcome Center for Human Genetics in Oxford. She has a DO chair, the Sir Henry Dale Fellowship DO chair, and she leads the computational rare disease genomics group. And this is why we invited her, because she has made a career in using computation approaches to interpret the role of genetic variants in rare disease. And this is what she will present today. And she will in particular focus on variants which are in the non-coding part of the genome, or the non-coding exome, and how basically they are associated with traits and rare diseases. Nicky, the floor is yours. Thank you again for accepting our invitation.
Thank you very much, Alex. Okay. Okay, nobody tells me otherwise. I'm going to assume you can see my slides. It's great to be here. Thank you very much for the invitation for the second of these webinars. I'm really excited to talk to you about some of our work recently on the non-coding exome, and I'm going to touch on aspects of both diagnosis and also on therapies. So rare diseases, although in the UK they're defined as diseases that affect fewer than 1 in every 2,000 individuals, although being individually rare, are collectively incredibly common, impacting over 300 million people worldwide. And while we've got pretty good at finding diagnoses for these patients, currently only around half of all patients get a genetic diagnosis. But even for those that do have a diagnosis, over 95% of rare disorders currently have no specific and effective treatments, meaning that vanishingly few of these individuals are actually treatable. But what I'm going to try and convince you of today are that genomics data are powerful tools to inform both pieces of this, so to inform both diagnosis and also therapies. So I'm going to start for where my team has focused since its inception 5 years ago now, on this portion in the middle, so trying to increase the diagnostic rates in individuals with rare diseases. So our current approach in clinical genetics to try and identify the disease-causing genetic variants is incredibly protein coding focused. So what I'm showing you here is the Turing pipeline for the Genomics England project here in the UK, where for a variant to actually be considered as a cause of a patient's disease, it has to be protein altering in at least one transcript. And if not, it kind of gets chucked out immediately into this box of not tiered, so not considered. But there are many more important genomic regions across the entire genome that have roles in regulation, for example, where variants could be important. But the problem with this is that genome is massive, right? We have 3 billion base pairs of DNA and a hell of a lot of variation across that genome. So where do we start? So the title of this talk gave you an insight into that. So my team likes to focus on the regions of the genome that are exonic. So a lot of people, human geneticists, where you ask kind of, where you speak about exons, would assume that an exon is a region that codes for protein. But actually the human exome is much, much bigger than that. So protein coding genes have untranslated regions at the 5' and 3' end that are exonic but don't encode protein. And non-coding RNAs are also spliced. They have exons and introns and have non-coding exonic regions. And actually the definition of an exon is a region that is transcribed into RNA and remains in that RNA after splicing. And actually, if we look at what proportion of the human genome has different exonic annotations, shown here where the y-axis here is the total amount of human base pairage that has each of the different annotations along the x-axis, we see that the protein coding sequence, the bar on the left, is actually less than a quarter of exonic sequence in humans. But one important thing to note here is that what we like to refer to as whole exome sequencing actually captures very little of the human exome. So what I'm showing you here, the proportion of the bars that are coloured in are what is captured in the exome capture used for the UK Biobank. So as you can see that over 90% of the coding sequence is captured in by this technology, we get very little of anything else. So if we actually want to look for variation, first to say, why is this important? So this is a plot showing you flipping that chart on its side, where the bottom bar of this is humans, when then we have a stacked bar chart of each of these different annotations following the same colors as the previous slide. But what we can see is as we go through evolution to more complex organisms, including mouse and humans, while we don't get much variation in the proportion of the number of base pairs that correspond to protein coding regions, we get this huge expansion of the UTRs and also these non-coding RNAs, suggesting that for disorders such as things involved with brain development, for example, some of these non-coding exonic regions could be very important and human-specific. So in the UK, we're really fortunate that we have a wealth of genome sequencing data. And our government a few years ago invested heavily in using genome sequencing as part of routine care. So an individual who is diagnosed with a neurodevelopmental disorder, for example, would most likely have genome sequencing very early in their care pathway. And the fact that we have such a large data set of genome sequencing data sets, so we've got tens of thousands of individuals with rare disease, this gives us the opportunity to find diagnoses in these non-coding exons. So I'm going to tell you one specific story that some of you may well be very familiar with, focused on a non-coding RNA that shows you the power of these data, not just Genomics England, but other large genome sequencing cohorts that now exist around the world. This story starts with an awesome student in my team, Yi-Yang Chen. So Yi-Yang came to me one day and said, I've identified this really interesting variant. This wasn't what he was looking at. He was looking at novel translated regions. But nevertheless, he came across this variant. And he said, this is really interesting, right? We found this in 46 different individuals in the Genomics England project. And for 40 of these, this was shown to be de novo. So this variant was found in the affected child, but not in either of their parents. Now, I will admit that I immediately said to Yang, this is likely a sequencing error. I don't believe this at all. This is not the amount of recurrence that we expect for a de novo variant. But nevertheless, Yang was somewhat persistent. He said, hang on a minute, all of these individuals have as yet undiagnosed neurodevelopmental disorders. And in total, this comprised about 0.5% of the currently undiagnosed neurodevelopmental disorder individuals within Genomics England, of which there are just shy of 9,000. And we couldn't find this variant anywhere else. It wasn't in around 50,000 individuals in Genomics England that are either unaffected or have disorders that are not neurodevelopmental disorder related. And in the population controls, it's absent from NOMAD, it's absent from all of us. And it's in one annoying individual in the UK Biobank. We still don't know why. My assumption is that it's somewhat mosaic across tissues, although we can't prove that. But nevertheless, despite that one annoying individual, we had an odds ratio of 2,500, which is pretty crazy in the rare disease world. But when I really believed that this was true and not some kind of sequencing artifact in Genomics England was when I then reached out to a collaborator and friend, Anna Donna-Laurier at the Broad Institute, and said, can you please look in your rare disease cohorts and see if you find this variant? And this response I got saying that I just discovered a new disease gene and diagnosed 8 of their families. Rather kind of serendipitously came through on the 29th of February, the rarest day of them all and Rare Disease Day, which I still think is quite cool. So what is this variant? So this variant exists in the middle of the ReNU42 gene, which is a small nuclear RNA. This gene is 141 or 145 base pairs long, depending on if you trust GenCode or other organisms and the conservation. I think it's actually 145. But this variant exists in the very center of this RNA, shown here by this tower of triangles on the top of the plot. And what we noticed is when we looked at variants in the general population, plotted on the bottom of this figure, we noticed this variant existed within a wider region which corresponded to about 18 base pairs, where variants were severely depleted in the general population. And when we looked in this wider region, we actually found additional variants, none anywhere near as common as our first variant we identified, but we found additional variants within this region in individuals with undiagnosed neurodevelopmental disorders. And this was a case across other different cohorts when we looked at through the individuals in GREGOR and the Undiagnosed Disease Network in the US, cohorts in Australia, and also matched with Christelle Depien and her team through Matchmaker Exchange. And altogether, we brought together a cohort of around 115 individuals with variants in this very small region. So I'm not clinically trained at all. So in order to try and bring together the phenotypic data associated with this patients, we worked with the wonderful Anne and Gabrielle and Sarah and her team. And initially we had 49 individuals where we had deep phenotypic information. And we showed that all of these individuals had a global developmental delay, with the majority of this being severe. They all had intellectual disability, and the vast majority of them also had microcephaly, was short in stature. Most of them were non-verbal. They had issues with vision and feeding, and also a lot of them had seizures. And the majority of these individuals actually had involvement of multiple different organ systems. There is also, I'm told, by dysmorphologists, a very clear facial gestalt, with individuals with variants in ReNU42 having very similar facial features. So the thing that still completely blows my mind about this discovery is an idea about the frequency. Now, it's incredibly hard to actually define the frequency of variants in ReNU42 for all-cause NDD. And the reason for that is nowhere in the world do we have the case where we are genome sequencing every individual that walks into a clinic presenting with an NDD. So we don't have an unbiased cohort. As such, we kind of need to make estimates around these numbers. But in Genomics England, we have 60 of our just shy of 9,000 undiagnosed NDD probands have variants in this 18 base pair region, so about 0.7%. What we then did is we used the Deciphering Developmental Disorders Study, or DDD project, to try and estimate how many individuals we would expect to be diagnosed before we got to our undiagnosed cohort in GEL. And in this article from the DDD study, their diagnostic rate is 40%. So if we assume that 40% would be diagnosed upstream, this would estimate that variants in ReNU42 would cause about 0.4% of all neurodevelopmental disorders. That's a bit of a pointless, meaningless number, so let's compare it to some of the genes we know and love. So here are the 3 protein coding genes that contribute the most diagnoses in the DDD project, the leading one being ANCARD11 at around 0.7%. Now, these numbers are not too dissimilar from the number for ReNU42. But what is particularly striking is that all 3 of these protein coding genes are in excess of 2,300 amino acids long. That's 7,000 base pairs of hit space for de novo variants to occur in these genes. And that's compared to an 18 base pair region of a non-coding RNA. So I think this is pretty damn remarkable. So hopefully I've convinced you that this RNA is important. And variants in it cause a severe neurodevelopmental disorder. But why? What on earth does this non-coding RNA do? So RNU42 encodes U4, which is a small nuclear RNA component of the major spliceosome. So the spliceosome is a complex of proteins and RNAs that is responsible for removing introns and the splicing together of the exons during mRNA processing. And the precise role within the spliceosome of U4 is to recognize the 5' splice site, so the left-hand end of the intron, and tell the spliceosome exactly where to splice. So here is an image from cryo-EM showing one of the complexes of the spliceosome, the U4, U6, U5 tri-SNP. And U4 is represented in the middle of this in red. And the U4 snRNA is tightly complex to another snRNA called U6. And these two snRNAs need to be unwound for the activation of splicing. So the variants that we find in individuals with NDD map to two distinct regions in this U4, U6 duplex that needs to be unwound. And these are in the middle as on this U4 that's shown here in teal. And on the right-hand side, you can see a zoom in of the structure of these two snRNAs. And these variants, the single nucleotide variants shown by bases in orange, and the single base insertions we identify shown by arrows, they exist in two distinct structures. One at the top here is the stem3 structure, and at the bottom we see this two-loop or quasi-pseudoknot structure. And the role of these two structures is thought to hold in the correct position this U6 agar-agar sequence, shown here as this flexible linker sequence. And that is the sequence that directly binds the 5' splice site in the activation of splicing. So with this hypothesis that these variants might disrupt this 5' splice site selection, given the role of these two regions, we looked at some RNA sequencing data that we had for 5 individuals with RNU42 variants within Genomics England. And this was data analysed by the wonderful Ruby Dawes, a postdoc in my team, and Jenny Lord, who's a lecturer now in Sheffield. And what we looked at specifically were significant events detected by the Fraser-2 tool that corresponded to a shift in 5' splice site usage. So rather than splicing at the canonical site at the end of the exon, where we saw that instead splicing happened at alternative sites, either upstream or downstream. And as you can see on the left, we see the significant increase in these events that correspond to non-canonical 5' splice site usage. And the thing that we were particularly pleased to see is that this change in 5' splice site usage wasn't random. We actually saw the same events shared between different samples, where we saw exactly the same non-canonical sites being used instead of canonical sites across these samples, showing you that there was something systematic in this splicing disruption. To look into this a little bit further, we looked at the motif, the sequence surrounding these splice sites that were either increased or decreased in usage in these individuals. So on the bottom here is the sites that are decreased in usage, and actually this motif is pretty much the canonical 5' splice site motif. And on the top we have the sites that are increased in usage. And hopefully you can spot some differences, specifically at the +3, +4, and +5 positions, where we see an increase in T at +3, an increase in C at +4 and +5. And what was really interesting to us was that these 3 positions are the exact 3 positions of the 5' splice site that directly bind to the U6 agar-agar sequence during the activation of splicing. So this really beautifully joined up this mechanism for us and gave us some evidence that this was what was driving this disorder. So Christelle Depienne and Caroline Neva produced this wonderful follow-up paper that is as a preprint but has been accepted in Nature Genetics, where they amassed an even larger cohort than we had in our first paper and did further phenotypic characterization of individuals with variants in RNU42. And one of their key findings in this paper was actually variants that exist in these 2 substructures, this stem3 region and this T-loop or quasi-pseudoknot, differ in phenotypic severity. So on the right-hand side is a PCA plot where phenotypes are ranked as 0 or 1 based on being a milder or a more severe form of a particular phenotype. And you can see that these stem3 variants cluster to the bottom left and are associated with a milder phenotype. And even more interestingly, what they then showed with a greater number of individuals with RNA sequencing data was that this severity of the phenotype actually correlated with greater disruption to 5' splice site usage. So they saw more disruption to splicing in individuals shown here in red with severe phenotypes and variants that are in the T-loop structure, compared to the milder variants, which look much more like the control variants, which are shown in purple. Another finding that we were perplexed about in our initial paper, but was also replicated by the Naver et al. Paper, was that we find that the de novo variants that we identify are almost exclusively derived on the maternal allele. So using nearby variants, we can phase these variants and figure out which allele they arose upon. And actually in our study, every single one of them was maternally derived. And in the Naver et al. Paper, they actually had some milder variants that actually were paternally derived, but nevertheless, over 95% across the two studies were on the maternal allele. So this is completely counter to our normal expectation that 80% of de novo variants would be paternally derived due to their increased cell divisions during spermatogenesis. And we really don't know the reasons for this result still. But one of the most interesting hypotheses that came from a conversation on Twitter was that nuclear RNAs during spermatogenesis actually are some of the only RNAs where selection happens on the haploid level because of cytoplasmic sharing of RNAs. And this might be a mechanism through which these mutations might actually be lethal in sperm. But the kind of crazy thing about that hypothesis is if this sole maternal derivation is being caused by a selection effect and lethality in sperm, the mutation rate associated with particularly that primary insertion that Yang initially identified, which we find in about 3/4 of all patients, would be even higher than we were already observing, which is a little bit crazy. So this has been a bit of a journey for me and my team. For the preprint out about this time last year in April, and then in July, our paper was published in Nature, and there was this associated press release from The Guardian. And as part of this, the wonderful Nicole Cedar, pictured here with her daughter, Mia Joy, gave us this wonderful quote. And as a bioinformatician, I think it's really beautiful. It says, "It's one thing to write papers and crunch all that data, then another to see a family with a precious unique child who is living it day by day. This is where data meets real life." They also said that they like to refer within their family to ReNU as ReNU, which is a really clever play on the genome, as their family is being renewed by this new information and hope for the future. And with the support of the families, we actually got the disorder actually named as ReNU syndrome. Owen first suggested this terrible acronym of different features of the disorders as NEDHAFA as a name, but the families really, really disliked this and very strongly voted for it being ReNU syndrome. And I think it's really great that the families have chosen this, and it's a name that speaks to the positivity. So it's also been a learning curve for me. I've spent a lot of time doing introductory slides where I try and communicate to people the impact of a diagnosis on a family with a rare disease. And this has become very stark, and actually I now have some firsthand experience of the kind of impact this can have. There's a wonderful Facebook group on the left here set up by a mum called Jessica, who's based in New York. And it currently has 370 something members. I can't remember when I last updated this slide. But this is a space where these families can come together and they can learn from each other. And it's kind of really empowering to think about the difference that somebody with a 20-year-old can make to a family who are just kind of in the beginnings of this with maybe a 1-year-old. And they know that their child is still going to be alive in 20 years' time, and what their kind of puberty and teenage years looks like. We've also worked with Unique, who are a charity based here in the UK, to create a report, a guide for families on what to expect. And this is available online if anybody has any patients and would like to share it with them. A few months ago, I also spoke to the wonderful James Coney. So here on this picture is showing Charlie, who has ReNU syndrome, and his dad, James. And James happens to be a writer for the Sunday Times. And he'd previously communicated or written articles around claiming benefits for having a disabled child. And he really wanted to write an article about his family's experience of being diagnosed. If you haven't seen this, then please go and search for it. It's if you work in the space, because it's a really, really strong validation of why we do what we do. But two of the key things that they communicate, and most of the families have communicated, is firstly, a genetic diagnosis makes them know for the first time that it wasn't their fault. It wasn't something they did during pregnancy that led to this disorder. It was just complete chance. And as I said, this is now this Facebook group of allowing them to access support and to chat to each other. And they communicated that kind of finding all of these children who were like Charlie, Sarah and I and families like ours are no longer alone, which I think is really powerful. The families are completely incredible. Spearheaded by Jessica and her sister and many others, they've already set up a nonprofit in the US called ReNU Syndrome United. And they're actually organizing their first family conference, which will be in July. Hopefully I'll be able to get in the US to go and meet some of these families for the first time in New York in July. If anybody is working, there's a scientific symposium linked to the family conference, and you'd be very welcome to join us. The family group have been connecting each other and trying to figure out where they come from in the world, and this is a map that's available on their website. It's likely very incomplete, but these are the people that are known to the family group, with the colors of the points representing the variant. So these blue ones are all that kind of primary insertion variant that you first identified. And as you can see, this is a growing number of patients. This map makes me simultaneously very happy, but also very sad, because one of the things it really does highlight is disparities in diagnosis and access to genome sequencing. So unfortunately, the disparities associated with non-coding variants are even more stark than we have for other protein coding diagnoses, because to find them, we need genome sequencing. So there's a lot of work we need to do to increase access to these diagnoses around the world. Nevertheless, in various different countries around the world, this RNU42 has now been added to standard diagnostic workflows. So we're finding these diagnoses on a day-to-day basis. And our estimates, based on the numbers I showed earlier, are that there are likely to be around 100,000 individuals with ReNU syndrome worldwide, which is pretty crazy. Can take some water. So RNU42 is not the first of the small nuclear RNAs to be associated with disease. There are others, specifically RNU4 attack, which is the minor spliceome equivalent of RNU42, and also RNU12. With both of these being shown to cause recessive conditions. Given this known association, we want to look to see if there was evidence in Genomics England of any other snRNAs being associated with neurodevelopmental disorders. So we took 28 snRNAs that were known to be expressed in brain, and we looked for various different hypotheses that followed what we'd observed for RNU42. So we first looked for recurrent de novo variants like our primary insertion variant, but didn't find anything that was highly recurrent. We also did a burden test across the length of each small nuclear RNA, but didn't find them enriched for either de novo or recessive variants. And finally, based on the fact that we had this region in the middle of RNU42 that we could identify by depletion in the general population, we also did a very simple depletion scan. This was performed again by the wonderful Ruby Dawes, where she looked at the proportion of all possible SNVs that were observed in 18 base pair windows scanning across each of these genes. And we can find these regions that are really depleted of variation. And then we did a burden test in each of these smaller regions. But none of the others other than RNU42 had any significant association with neurodevelopmental disorders. However, one that we were particularly interested in was this one, RNU5B1, which came with the third most highly depleted region within the sequence. And we initially identified 3 individuals with de novo variants in this region with neurodevelopmental disorders, and no individuals without neurodevelopmental disorders had de novo variants in this region. And when talking to the wonderful Christelle Depienne, when they were preparing their RNU42 follow-up paper, she also said that they also had variants in RNU5B1 and also RNU5A1. And all of the variants that were identified by all of us were found within the same region, this conserved 5' stem loop region. And we now propose this as another snRNA associated with neurodevelopmental disorders. So this was also shown in another preprint from Sid Banker, his team, and Adam Jackson and his team who are in Manchester in the UK. And they, and also a team from Ernest Turrer and Daniel Green in New York, identified also RNU22P, which actually was annotated as a pseudogene, but is clearly, I think it's being re-annotated not as a pseudogene because it has a very clear role in neurodevelopmental disorders. So it's very cool that we're now identifying more of these genes that are associated with NDD and getting diagnoses back to patients. Also, interestingly, there was a very recent preprint, I think back in January, linking a couple of variants within RNU42 and also variants within the interacting partner U6 in retinitis pigmentosa. So in this plot, you see the central region with these underlying pink bases where we find variants in ReNU syndrome, and these two red variants around this loop region of recurrent variants in individuals with retinitis pigmentosa. So this is a really cool example of genetic pleiotropy. So I'm going to talk a little bit about some new data that we have here, which I'm really excited about, and we're very close to getting a preprint out on this. And this is an amazing collaboration with Greg Findlay, who is a group leader at the Crick Institute in London, and a postdoc in his team, Joachim. And they are really, really talented at doing saturation genome editing experiments. Greg and Joe made every single possible single nucleotide variant across the whole length of RNU42. Also, single nucleotide insertion deletions in the central region that we know is critical in ReNU syndrome. And they did this using a CRISPR approach into a haploid cell line called HAP1. And given that RNU42 is essential in HAP1 cells, we can then basically simply measure cell death and look how depleted variants are after a period of selection. So the variants that are more deleterious, there is more cell death, these variants are more depleted in the final assay scores. So the first result we find from this data, so on the left-hand side, what I'm showing you is this, what we call function score, where the majority of variants across the entire gene have a score of 0. This is where we normalize this to. And based on control insertions into loop regions, we defined a threshold of minus 0.039, shown here by this black dotted line, below which variants is significantly depleted in the assay. And what we're really pleased to see is that all the variants that we find in ReNU syndrome, shown here by these red points, were below this dotted line, so we're significantly depleted. And on the right here, what I'm showing you across the entire gene is the separate or the function scores of variants that were observed in the UK Biobank, or all of us, so in population controls in blue, and how these beautifully separate from the variants that we find in ReNU syndrome that are in red. With variants that are unobserved in either ReNU syndrome or the population controls, mostly being shifted to the right, so being not deleterious, but with quite a few of them also towards the right, to the left, sorry, with many of them being in this central region, potentially being variants that, if identified an individual with an NDD, could be diagnostic. We also, using this data, we defined the critical region that we'd initially defined for ReNU syndrome as 18 base pairs, shown here by these two vertical grey lines, as two much more specific regions of 9 and 4 nucleotides corresponding to the T loop and STEM3 respectively. We then split our variants into two based on their depletion score in the assay. So we drew another threshold at minus 1. This is a bit of an arbitrary threshold, but it gave us enough power, so enough variants in each category, where any variants with a score of less than -1, we said were strongly depleted, and variants between 0.39 and 0.1 we said were moderately depleted. And then working with Christelle, Caroline, and Elsa in their team, we redid their phenotype severity clustering and could show that variants that had these more moderate scores were corresponded to milder phenotypes, and again were clustering out compared to the stronger depleting, more severe variants that are shown in this plot. And to be a bit more specific, we saw that individuals with more strongly depleting variants were significantly more likely to have severe developmental delay and intellectual disability, were significantly more likely to be non-verbal or have only a few words, although we didn't find any difference in seizures. I think the starkest one here is so that individuals with more moderately depleted variants, 95% of them have normal speech or can at least speak in simple sentences. And this is compared to about 5% of individuals with more strongly depleted variants. So a really stark difference in this phenotypic severity. We also show that SGE massively outperforms in silico predictors. So there aren't many in silico predictors we can use to look at variants in non-coding regions, but CAD is one that is actually used that can predict a genome-wide. The problem with trying to use CAD to predict variant deleteriousness across things like RNU42 is that the entire gene is really highly conserved, which means there's pretty much no delineation in the CAD scores. They're all very similar. And actually, if you drew a CAD threshold that captured all of the ReNU syndrome variants, you would also annotate 60% of variants across the entire gene. So if we produce a ROC curve and look at the area under the curve for our SGE assay to separate ReNU syndrome variants from variants identified in the population, we get a really impressive area under the curve of 0.95, and CAD is actually pretty useless at doing this with a score of 0.65. The other thing we can do with these scores is to resolve variant pathogenicity. So there was this paper that came out very recently, so last month, proposing a role of variants in the 5' stem loop in dominant ReNU syndrome. So these two variants shown here on the bottom in this yellow region, and rather alarmingly in the initial paper, both of these variants were inherited from unaffected, I think, both mothers, whereas the vast majority of the variants that we find in ReNU syndrome are de novo. So that initially led us to the fact that these might not be real, but when we look at the SGE score, they score very much within the normal range. So suggesting that these variants aren't causing ReNU syndrome. Okay, time for some more water. So I'm going to shift gears a little bit here. So I've spoken about this middle box, so convincing you that genomics and particularly genome sequencing data is powerful to inform genetic diagnoses. But I want to talk a little bit about a newer direction of the team to think about genetic therapies. So we're in a really exciting space at the moment where there's been a huge technological advances, which are driving forward use of CRISPR-based editing technologies and also antisense oligonucleotides to treat rare disorders. And while both of these technologies can be used to target specific variants, so making single base changes to correct a specific patient variant or modifying, say, splicing to correct the defect caused by a splicing variant, for example, what I'm really excited about is the idea that we might be able to modify gene regulation on a gene level and create a therapy that would actually be useful for all patients with variants of the same mechanism, say loss of function variants across the same gene. Now, there are a whole bunch of different examples that support this hypothesis, whereby we have therapies that are either approved or going through clinical trials, where modifying gene regulation due to an in-depth knowledge of that gene regulation can be exploited to create an effective therapy. So in order to talk about this in the non-coding exome space, I'm going to focus on the kind of other favorite of my regions in the genome, and that is untranslated regions or UTRs. Now, my team has focused on these regions for the last 5 or so years. So for those of you who might not be so familiar, the UTRs are the regions directly up and downstream of the protein coding sequence of a gene, and they have incredibly important roles in regulating the stability of the RNA, where it's localized to within the cell, and the rate at which protein is actually produced from that RNA. And this regulation is mediated by a series of different sequence and structural motifs, and the binding of these regions to external factors, including RNA binding proteins and also microRNAs. So this is a figure that shows you a bunch of the different sequence and structural motifs that mediate that regulation. I love this figure because the coding sequence is just boring bit in the middle and everything else is shown in color. But in broad brush terms, the 5' UTR is involved in translational control, so controlling the amount of protein that is produced, whereas the 3' UTR, which is much, much longer, is involved in RNA stability and subcellular localization, as well as controlling translation. So I said my team have focused on these regions for a few years now. I don't have time to go into much of this in detail, so I'm just going to whiz through some key points of what we've learned over those years. So one of the first things we did, this is a paper from a wonderful student in my team, Nahama Wider, who showed that UTR-mediated gene regulation, there is differences in UTRs, particularly 5' UTRs, in dosage-sensitive genes. So genes that care about how much product they produce have longer, more complex 5' UTRs with more regulatory elements within them, hinting that translational regulation is really important. We've also done a whole bunch of work looking for diagnostic variants, showing that variants that disrupt UTR-mediated gene regulation are under really strong negative selection and can cause rare disease through a loss of function. Here is one example where creating an early start codon just upstream of the MEF2C gene that leads the ribosome to translate out of frame, overlapping the coding sequence, causes a loss of function and leads to a severe developmental disorder. So using that knowledge, we then did large-scale studies to look in Genomics England to find UTR variants that might be diagnostic. But our main takeaway from doing this, both looking at dominant de novo variants, but also recessive variants, was that these are not going to change the diagnostic landscape. We can create the tools to be able to find them, but we're not going to diagnose too many patients through these approaches. And just to briefly talk about one of those, this was a study led by Alex Martin-Geary, who's a postdoc in the team, to look at de novo variants in these regions. And looking at a set of 8,000 diagnosed probands, we found 11 candidate variants. 4 of these were already known and 5 likely new diagnoses. And to do this, we did so much bioinformatics work. We tried to annotate with every different predicted mechanism that we could think of across the UTRs and also an upstream promoter region, and still did not find too many new diagnoses. So we've done a lot of work to understand these regions and their role in disease, but can we use what we've learned here about gene regulation to inform therapies? So the hypothesis here is based around upregulation of haploinsufficient disease genes. So if you imagine a scenario where in the healthy state, there are 2 alleles of a gene that each produce 2 units of functional protein. In the disease state on the bottom left here, a loss of function variant in one of these alleles means that only one allele produces 2 units of protein, which is insufficient. But what happens if we could block or edit a negative regulatory element in the 5' UTR of the remaining wild-type allele? Could we use this to upregulate downstream protein production and recapitulate the normal amount of protein? So an element that is a negative regulator of translation that we've studied in some detail are upstream open reading frames, or uORFs. So these are start codons followed by stop codons that exist within the 5' UTR that disrupt the scanning ribosome. So the ribosome joins the 5' cap of the RNA and it scans until it hits an ATG, at which it makes a decision of whether or not to start translation. And these upstream reading frames are translated and they limit the number of ribosomes that make it downstream to the protein coding sequence. A paper back from 2009 showed that a handful of examples conferred a 30 to 80% reduction in protein levels, so they can have a very large effect. And they're not rare. They're actually predicted to occur in about half of all protein coding genes. So there are a couple of papers that had hypothesized that using an antisense oligonucleotide targeted directly to a uORF, you could block uORF translation and upregulate downstream protein production. But more recent work has shown that this does not reproduce. And I know multiple people have tried this approach across different genes and are struggling to get this to work. However, could we use a different approach? So we know that splice switching ASOs, so ASOs that target splicing regulatory elements and then modify exon inclusion and exclusion into a transcript, underlie many different effective therapies, including spinal muscular atrophy and Batten's disease, for example. So could we use an exon-skipping approach to modify uORF inclusion in transcripts as a treatment for rare disease? So imagine this scenario where we have a uORF, so a start codon of a uORF on the central exon of a 5' UTR, where there are upstream and downstream exons that are also form part of the 5' UTR. Could we then use a splice switching ASO to selectively exclude this central exon and upregulate the downstream protein production? So a super talented PhD student in the team, Eloise, has been looking at this. And if we look across main transcripts, we can see that there are around 1,500 main transcripts without at least one skippable 5' UTR exon. If we then use ribosome profiling data, which is a technique to detect active translation, we can see that just shy of 900 of these genes have a start codon on that central exon. And 66 of these are neurodevelopmental disorder genes. So I'm just going to show you one example where we've done some functional validation work, and this is the gene GRIN2B. So here I'm showing you the luciferase reporter assay where we clone mutant and wild type 5' UTR upstream of the luciferase gene and measure downstream translation. So on the top, I'm showing you the wild type 5' UTR containing the uORF and the central exon. In the middle, I'm showing you a construct missing that central exon. On the bottom, I'm showing you that construct with the central exon, but with all of the start codons in that central exon mutated. And here we can see that compared to the wild type 5' UTR, we see this dramatic upregulation of luciferase translation when we remove that central exon, and a similar amount of upregulation when we remove the start codons. The slight issue with this is, at least in the case of GRIN2B, this works a little bit too well. So here we're seeing a 7 to 8-fold upregulation of translation. And GRIN2B, unfortunately, is a gene that has both a loss and gain of function effect. So this works a little bit too well in this scenario. But nevertheless, there are other genes where this is a much more the kind of upregulation that we would desire. So one of the key issues in this field is how do we then make this scale? So we can identify individual disorders where we can create therapies. But one of our big challenges is to tackle the 4,500 or so rare single gene disorders that are known. And very much what I was trying to convince you of here, that there are ways of using genome or omics data, including the example I just showed you of exon-skipping uORF therapies, that allows us to predict what we might target and use genomics data to scale therapy, or at least target identification. So we know that genetic therapies can be reprogrammed. We have a single, a nucleotide sequence that forms either an ASO or the guide RNA for CRISPR. So we can create kind of an informatics problem here where we can predict the sequence that might be effective in any given situation or for any given gene. And this is something that we're really interested in doing in Oxford, and we're really excited also to collaborate with other people who might be interested in this space. And we've just had a recent influx of funding from the MRC to create a Centre of Research Excellence in Therapeutic Genomics, where we define this therapeutic genomics concept of using the application of the principles and technologies and approaches used in genomics to accelerate this therapeutic development, to accelerate the identification of targets, and to create therapies for more individuals with rare disease. So this is the group of individuals that are involved at the moment. I'd say we're really, really collaborative, but it brings together a whole bunch of people who are really excited about this place and doing this space and doing really interesting science. So just to circle back to ReNU syndrome, so clearly having identified this new disorder, we're also really interested in trying to develop therapies. There's various reasons we think that RNU42 is a really tractable target for developing therapies. It's in particular the fact that it's highly prevalent, so there is pharmaceutical interest here. It is very severe, and also the fact that there are a large proportion of diseases caused by that one single insertion variant means we can design therapies that either target the entire gene or that target that specific variant. But a slight caution that we're still very early days in our understanding of ReNU syndrome, and there's lots of things we need to better understand. So we need to have a better idea of mechanism and specifically the impact of either increasing or decreasing expression of RNU42 and its related homologs. We need to know more about the potential history of disease, so to try and figure out if we can actually make a difference to these patients through therapy development. And we need to find effective biomarkers. And for that, we're doing a lot of work trying to develop cell and mouse models that might be able to help us try and find biomarkers, although we're buoyed by the fact that there is an obvious splicing signature here. So just to conclude, so hopefully I've convinced you that genomics data are powerful tools to inform both diagnostics, but also therapies. It's really exciting to see the role of non-coding RNAs as a really evolving field. And I'm excited to see how many of these RNAs we find to be involved in rare disease. I think a data-driven approach can help to scale therapy design to help us bring more therapies to more rare disease patients. But for us to make any progress in this field, both in diagnostics and in therapies, data sharing and collaboration globally are really, really key to driving progress. And I do believe that we can create a world where one day a genetic diagnosis will actually lead to a therapy. So just to acknowledge all of the huge numbers of people that are involved in the RNU42 work, and of course to acknowledge the participants and the clinicians in the recruiting centres. Without them, none of this work would be possible. This is my wonderful team doing Secret Santa in December, and also to thank all my funders, and thank you all very much for listening.
Many thanks, Nicky. So please use the Q&A possibility to ask questions. And basically to organize the discussion with Nicky. And maybe before I get those questions, maybe something you said that most are de novo mutation in ReNU42, but you said most. And so what is the phenotype of transmitting parents when this is the case?
So I think there's only 1 or 2 examples of a milder SNV, and I guess we don't know if they are fully heterozygous in those parents or if it might be mosaic. I don't think we have enough to really look at that very much. And there's definitely one where it's inherited from an apparently unaffected mother, but it's also in the kind of more recent NHS data where our phenotyping is a little bit more suspect. So it needs diving into.
And a bit a related point of view, you said that there is submitted a preprint linking the partner RNU6 to retinitis pigmentosa compared to RNU42 being, let's say, a classical NDD. So how do you reconcile this, you know? I mean, it seems that the splicing, if you mutate one, splicing of something in the eye is not correct, and you go to RP, while in the other ones, I don't think that they have major high vision impairment, basically.
So I think it's expression driven. I could be wrong. So actually the RP for the U6, they find this across the 5 different U6 homologs, paralogs. I get confused between those 2 words, sorry. But they've each of which are somewhat equally expressed or expressed to a significant level. Whereas for RNU42, it's significantly more higher expressed than RNU41, which is the next most highly expressed of the U4s. So I think it's because RNU42 is just the primary gene, whereas in U6 there's much more compensation happening.
So there was one question on this, and you immediately answered. So do you expect 41 to be a recessive disease? If we go even further?
I don't know. Possibly. I don't think we've looked for recessive variants in the ReNU region in 41. Don't know.
When you did the MAVE, both, maybe I missed it, both alleles were edited, or it was an haploid cell lines, and you were basically looking only on one allele?
It's a haploid cell line. So
okay, an haploid cell line. Because there was, we had other question about, eventual component zygotes and an extremely severe phenotype, but you haven't seen this, obviously, and not even in your MAVE essay.
So we have some interesting variants that might follow a recessive phenotype outside of the critical ReNU region, but we're still following that up at the moment.
And some of our listeners basically ask, is there a tool or browser when, I can use your MAVE result, and so your SGA scores is in a diagnostic setting?
Very soon.
Very soon.
The preprint is just going around the authors at the moment for approval, and when we release that, we will release the scores. So then you'll be able to use the data. So hopefully in the next couple of weeks.
And then, if we go further, I mean, many people have asked, if basically going back to the therapy, and so what happens if you have a protein coding genes and you don't have a micro-HORF you can play with? What do you suggest? I
think there's lots of different approaches. Right, there was a preprint that appeared, was that this week, from Lezenkovicis and co, that were looking at lots of different mechanisms to try and predict what proportion of haploinsufficient disease genes would have therapies that could be looked at. But There's quite a few different approaches we can think about this. So poison exons is another. So looking for naturally included poison exons that lead to unproductive splicing and try to limit their inclusion to increase the amount of functional transcript that is produced is obviously another viable approach. So I think we're going to kind of have to categorize all of these different genes into which approach is going to work for each specific gene, which is, yeah, it's going to be fun.
So, and you gave us an example where it was working too well.
Yes.
You know, did you try to tease it apart a bit further? You know, adding pieces, you know, you just took out one, you know, the ATG of the, you know, did you try to play a bit further?
We've not teased that one apart in too much detail because we then just realized it was probably quite a bad idea because of the gain of function phenotype. It's a really nice example of the fact that we can upregulate. And one of the nice things about ASOs is you could titrate the dosage. So in theory, you could use a small dose of ASO and you might be able to have a smaller upregulation, but it's quite a dangerous thing to try in a patient. And also, we're doing a luciferase completely out of context assay here, and we don't know if that's the amount of upregulation we would achieve with an ASO against the endogenous locus, which is something else that we're trying.
Yeah. And I have one, actually, something you mentioned, and you said, so you look at the genes which were affected when ReNU4 was mutated. So can you pinpoint to certain genes that might be playing a role in the phenotypes?
So we've not looked at that in too much detail yet. The reason for that is the data that we have and that we initially analyzed was all blood RNA sequencing data. I also think we need really, really high depth RNA sequencing data to look at this. So this is a really subtle effect. Obviously, global disruption to 5' splice site selection would be embryonic lethal. So it's a really, really subtle signature with subtle shifts in 5' splice site usage. So some higher depth sequencing data would be better to kind of tease that out.
And I have a new question going back to therapy. So given that you target genes which are expression sensitive, one may assume, and you said it, that overshooting can also be bad and deleterious. So, and the extent of overexpression may vary between individuals. So how do you plan to address this issue? You know, how will you control for variation in the therapy, basically?
That's a good question. I'm going to bow out of it and say that I'm the bioinformatician very early in the process and somebody else figure out the complexities.
Fair enough. Fair enough. Okay, I think that's I've been through the different question. I mean, please, if there is a pressing question for the audience, please write it in the Q&R. If not, I mean, Nicky, we want to thank you and your team for the beautiful work presented today. And with this, I have just congratulations for you coming, so, which I think is a good point. Maybe I should also point that we were more than 200, and so thanks to you, Nicky, thanks to Bill to come with the idea of organizing those webinars. And I think we have a public, and so I think it's great. Thanks to everybody.
Thank you for having me.